Good afternoon, dear students. In the last lectures, we were studying regarding downstream processing or product recovery in a fermentation industry. Okay, there are various steps involved in downstream processing. So, we have discussed some of the steps, like we have discussed number one, cell harvesting, number two, disruption of cells, number three, extraction of products, and number four, concentration of products. Now, today we are going to discuss about purification of the product. See, purification of the product can be done by chromatography. It can be done by crystallization. It can be done by dialysis or electrodialysis. Okay, it can then it can also be done by distillation process. Right. So now let us discuss about the first method of purification most commonly practiced, that is chromatography. Now, the biological products of fermentations like proteins, pharmaceuticals, diagnostic compounds, and research materials are generally effectively purified by the process of chromatography. What is chromatography? It's basically an analytical technique that deals with the separation of closely related compounds or separation of components that are present in a mixture and the components are closely related. Now, chromatography usually consists of stationary phase and mobile phase. The stationary phase is generally a solid or a liquid, okay, or it may be in this case during purification it is a porous solid matrix that is packed inside a column, okay, and it is equilibrated with the solvent, and the mixture of compounds to be purified is loaded on it. The mixture of compounds to be separated is loaded on the column packed with the matrix or solid, rather you can say a stationary phase. The compounds are eluted. The compounds present in the sample mixture are eluted or separated out by a mobile phase. The mobile phase is a moving phase. It moves over the stationary phase. Now, different types of chromatographic techniques used for separation are gel filtration chromatography, ion exchange chromatography, adsorption chromatography, the most commonly practiced ones. Now, the first one is gel filtration chromatography. It is also known as size exclusion chromatography because separation is done on the basis of size, shape and molecular weight. Rather, you can say separation is done on the basis of molecular size. The sponge like gel beads in gel filtration chromatography, gel beads are the stationary phases which are filled in a column. They are packed in a column. The sponge like gel beads have pores. They serve as molecular sieves for separation of smaller and bigger molecule. A sieve is like when you want to when you prepare tea, okay, you sieve it, okay, so as to separate the solid tea particles and to separate the liquid tea, okay. So like that, the sponge like beads are porous. They have pores on their surface. The pores serve as molecular sieves for separation of smaller and bigger molecules, okay. Smaller molecules cannot pass into the pores of the bead, so they get separated first or easily. Larger molecules cannot enter inside the pores, so they come out of the column very fast, or they come out first. The smaller molecules can enter inside the pores of the gel bead. Small molecules enter inside the pores of the gel bead, and hence they are eluted late, or they come out very slowly, right? This is how separation is done by molecular sieve method or gel filtration method. The solution mixture containing various components of different size is applied on the column and they are eluted out. Smaller molecules enter the gel bead through their pores and get trapped inside the gel bead. On the other hand, larger molecules cannot pass through the pores and therefore come out first along with the mobile liquid or mobile phase. At industrial scale, gel filtration is particularly useful to remove salts, low molecular weight compounds from high molecular weight products. Okay. The second is ion exchange chromatography. It involves separation based on surface charges. Okay. So ion exchange chromatography separates molecule on the basis of the charge they possess on their surface, or the molecule can be made to acquire charge by altering the pH. There are two kinds of ion exchangers, cation exchangers and anion exchangers. Cation exchangers have negatively charged, you can say, functional groups, and anion exchangers have positively charged groups like DEAE. Okay. In ion exchange chromatography, the pH of the medium is very crucial, and pH determines the effective charge that is present on the target molecule as well as on the ion exchanger. So the molecules which are bound to the ion exchanger, when you load the sample on the ion exchanger, the ionic molecule, that the your product having particular charge, will be uh, bound to the ion exchanger, rest of it will flow out. So the ionic bound molecules can be eluted out from the matrix by changing the pH of the eluent or by increasing the concentration of salt solution. Okay. So your product which is bound to the ion exchanger is eluted out by changing the pH of the eluting solvent or by increasing the salt concentration. Ion exchange chromatography is useful for purification of antibiotics and some proteins. Next is affinity chromatography. It's a very elegant or a soft method for purification of proteins from a complex mixture. Affinity chromatography is based on interaction of a protein with the immobilized ligand. If your protein product is having affinity with the ligand, it will attach to the ligand. The ligand is immobilized inside the column. The ligand, what is a ligand? It can be a specific antibody which will bind to an antigen, or it will be a substrate which can bind to its enzyme, or a substrate analog, or it can be an inhibitor. The immobilized ligand on a solid matrix can be used to fish out complementary structures. Okay. Or to remove or to extract out the required product. The protein bound to the ligand. Now your protein, if you want to separate or if you want to purify and you load the sample, and your protein product, pure protein product, is bound to the ligand and rest of the impurities have come out. So now the protein that is bound to the ligand is eluted out by reducing their interaction. The two have affinity for each other, so they have attached to each other, they are bound to each other. Now you reduce the interaction between them, which is achieved by changing the pH of the buffer, or altering the ionic strength of the buffer, or using another free ligand molecule. The fresh ligand used have to be removed in the subsequent steps. So the protein bound to the ligand can be removed by one or the other way, and you get a purified protein product. By crystallization also, there are certain products which are obtained in crystalline form, crystal, solid, small solid particles. So crystallization can also be a method of purification. This method is used for purification of low molecular weight compounds like antibiotics, citric acid, etc. Say for example, penicillin is purified by crystallization process. Okay, by using various salts. The products can be crystallized by evaporation, or by low temperature treatment method, or by addition of certain organic solvents. Okay, or that is chemicals also. By addition of chemicals or organic solvents that help the solute or help the product to undergo crystallization. The solubility of the product is reduced. We reduce the solubility of the product by adding solvents or salts or polymers or polyelectrolytes, or by altering the pH. We reduce the solubility of the product so that it undergoes crystallization. Decrease in solubility causes it sometimes to precipitate or to crystallize, and the precipitate can then be extracted, and then crystallization can be done by using various chemicals. Distillation can also be a method if we want to recover volatile solvents, like if you are recovering ethanol or acetone or butanol, then distillation is the best method used for recovering the volatile solvents. The fermented broth is subjected to heating in coiled pipes which is present in the rectifier column by ascending hot vapors produced from the analyzer column, and then it passes through the analyzer, and depending upon the temperature at which they evaporate, they can be extracted out. The finishing steps, the last process, the last step involved in the downstream processing. Most of the time it is, sometimes finishing is done by crystallize, taking out the product in crystalline form, that is crystallization can also be a finishing step, or the most important finishing step in most of the cases is drying. If your product is to be obtained in powdered form, not in a liquid form, then we have to subject the product to drying. So drying is supposed to be an important or essential, you can say, step involved in product formulation. Okay, it basically involves transfer of heat to the wet product for removal of moisture. If your product is wet, even if the crystals or precipitate is wet, you have to remove the moisture. Okay, so you apply heat to remove the moisture, or sometimes you have to apply very gentle heat also to remove the moisture. In certain cases heat cannot be used, we'll discuss now. Okay, so the wet product is subjected to a treatment which will convert it into a dry product by removing the moisture. Most of the biological products of fermentation are sensitive to heat, and therefore require very gentle drying method. Now, based on the method of heat transfer, drying devices may be categorized as contact devices, convection devices, or radiation dryers. Where we cannot apply heat, we have to apply other methods. So, for example, spray drying. Spray drying is used for drying large volume of liquid. If you have large volume of liquid, you have to spray dry. Okay, drum drying. Say for example, if you want to produce milk powder from milk, so through a narrow nozzle, the milk is sprayed in the form of a mist, okay, or in the form of fine droplets on the surface of rotating drums. And as soon as the milk is sprayed, the drums are heated and the milk immediately gets converted into dried powder, which can be removed or scraped off by knife. So, if you want to dry large volume of liquid, spray drying is practiced. In spray drying, small droplets of liquid containing the product, okay, you can say that liquid is sprayed or in the form of a mist through a narrow nozzle, directing it over a stream of hot gases. Okay, so the liquid is sprayed towards from a nozzle in a direction where there is a stream of hot gases. The the water immediately evaporates and solid particles are left behind, which can be collected. Rotary drum dryers remove water by heat conduction. The drums are heated. A thin film of suspension of the liquid product is sprayed, or the liquid is sprayed over the heated surface of the drum. The drums are rotating constantly and you are spraying your product on it, and the product dries immediately because the drums are heated. The dried solid product is scraped off with a knife and collected. We also have vacuum tray dryers, in which there are small trays where we keep our product. In vacuum tray dryers, material to be dried is placed on heated shelves within a vacuum chamber. Here the chamber is not having any air, it is evacuated, and the chamber is having shelves. There are trays in the shelves. Okay, so you keep your product in the trays and the shelves, and they are kept in a vacuum chamber. This allows drying at a very low temperature because you are drying in vacuum. So this allows lower temperature to be used because boiling point of water is reached at reduced pressure, because there is vacuum now. So at reduced pressure, boiling point is reached, because okay, lower boiling point of water is reached. This method is used for small size materials like pharmaceutical products or heat sensitive products. Freeze drying. Freeze drying is also called as lyophilization. Okay, in case of lyophilization, this is the preferred one if you want to dry your microorganisms, vaccines, heat sensitive material, vaccines, okay, viruses. So bacterial cultures, they can be freeze dried. Okay. Most preferred method for wide range of products like pharmaceutical products, certain foodstuffs, diagnostics, bacteria, viruses, vaccines, etc. This is mainly because freeze drying usually does not cause loss of any biological activity of the desired product. The biological activity is not lost. So vaccines, virus, bacterial culture, diagnostics, pharmaceuticals, certain drugs, they are dried by freeze drying. Lyophilization is based on the principle of sublimation of a liquid from a frozen state. In the actual technique, the liquid containing the product, product in the liquid form is frozen, and then it is dried freeze dryer under vacuum. So the liquid is actually first frozen, it is freezed. After freezing, it is dried in a freeze dryer under vacuum. The vacuum can now be released, and the product containing the vials are sealed. So immediately the product is freezed, and then it is freeze dried and converted into powdered form. Under vacuum. So there is no rise in temperature at all. The vacuum present in the product can be released. And the vials are sealed. For example, penicillin, antibiotic penicillin is freeze dried directly in the ampules in which it is taken. So that is all about the various steps that are involved in downstream processing. So with this we finish the downstream processing.